Again, greetings class. This is a presentation in the 4th series, and we are moving reasonably well. I recommend that you go sequentially according to the series of the presentations so that you are able to follow the story in completeness. Today, we are going to be looking at a progression from characterization or screening. Remember, screening is the evaluation, and you have done the evaluation of your particular lines. You have the data, you have done some statistics, and you know the way out in how you can now handle the accessions of promise and crossing. So we want to go from that step to the reproduction, and that's what I'm calling the R system window. Remember the ad humor. That window should open up the crossing or the hybridization exercise. And so the reason why I am suggesting strongly that we follow the progression, we have moved from the assessment towards desired features. This is where, in fact, that bit is the basis of the farmer participatory breeding. They tell you, as the consumers, what kind of the product target that the target product that you are endeavoring to produce, to develop for them. Then we looked at the issues of developing breeding goals and having a plan. And then from there, we spent considerable time and focus on assembling the choice germplasm, characterizing and evaluating, and then doing some of the statistics, examining the statistical options that can inform on the identification of the accessions of promise to actually go on with crossing the parents. Remember, we have an interest of identifying a number of parental lines that can go into a crossing. As I mentioned before, our intention as breeders is to cross good parents with good parents. And then we, at this point here, we are looking at studying the reproduction, but we brought this reproduction a little beyond, because we've already talked about creating experimental design. We have looked at the augmented design as a field layout design. We have looked at the limited aspects of the quantitative genetics to operationalize the breeder's equation. In fact, we'll be moving into that if we have not done so. And we will then look at a few other things that follow this sequence in this table, in this scheme. It's very important to keep track of it. So we want to look at the importance of plant reproduction systems of important crops for defining self and outcrossing breeding strategies. On completion of this unit, you must know the following things. I do not want to read through them because you'll see them on the slides that will be uploaded. Now, I need to emphasize the primary purpose of focusing on the reproduction of plants. One is to create by hybridization variability more rapidly. We have other ways of creating variability. We can irradiate materials to create new variability. We can hybridize to cause new variability. We can actually do tissue culture and induce soma clone variation as a way of hybridizing, uh, of creating the hybridization variability more rapidly. However, we are going to focus on the issue of crossing. It's really a mimic when a plant breeder does that, of what the plants themselves do to actually cross among and between themselves under natural conditions. When we do the crossing, we manipulate manually. That is called enforced or artificial selection. Now, reproduction focus is also important as it determines a breeding method. Extremely important because we have various breeding methods. And unless we can understand the flowering, the reproduction, it can be very difficult for us to design a proper breeding method. Then also, the reproduction focus determines seed multiplication and maintenance procedures. Remember, the breeder's task almost will stop at the breeder seed. And that breeder seed is so limited that it needs to be multiplied further for distribution to farmers. That's quite another process. But here, the seed multiplication, the maintenance procedures that include isolation and the emasculation and stuff like that is dependent on the understanding of the reproduction focus. And then, of course, as we mentioned before, associated with the breeding method is the development of a cultivar type desired. For example, we have a common talk of the OPs, open pollinated varieties, versus self lines, etc., etc. So the first port of call in this is to begin to look at the flower morphology as a basis of the hybridization biology. And I'll move fairly quickly on this one because it is described as we go further in the slides. I'll not go again part by part as we'll have to understand this in the subsequent slides that we'll be talking about. And of course, you'll have this in your notes. What I want to show here very quickly is really the enormous diversity in the flower colors, in the flower morphology. They are just amazing. They are just amazing, 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 amazing. So really the astonishing reproductive diversity of flowering plants has attracted the senses and curiosity of humans since the dawn of civilization. As we know, till today, flowers have a long played prominent role in religious ceremonies. And they are also subject of countless artistic endeavors. You can note the many funeral ceremonies in which flowers are presented to the sick in hospital, presented in ceremonial celebrations of remembering those who have departed from us. But above all else, particularly in the context of plant breeding, understanding plant reproduction is of immense practical importance. In plant breeding, for example, flowers are the port of call for artificial selection by way of making crosses between parents of promise. Remember the saying, as I've already mentioned, crossing good with good of the identified germplasm accessions from characterization and or evaluation. The importance of flowers is expressed in the slides that have already been made available to you and the ones we have seen and will see. So the key question is this: why should the reproductive structures of flowering plants, and remember flowering plants are angiosperms, to exhibit such an extent of variation than any other group of organisms? This question is particularly surprising when one considers that the flowers just serve one main function, and that function is to promote mating, the natural crossing, the natural hybridization. Now, the answer is in the reality that plants are not as mobile as humans. They must engage the services of pollen vectors to ensure that cross-pollination and the offspring of high genetic quality is realized. Rather than relying on wind alone or a group of animal pollinators, the reproductive structure of a majority of flowering plants have undergone spectacular waves of diversification. Pollinator-driven speciation has played, indeed, a very important role in the evolution of angiosperm diversity. And it is in that respect that we see flowering plants displaying spectacular floral diversity and a bewildering array of reproductive adaptations that promote chiefly mating, particularly for outbreeding species. A structure or a striking feature of this diversity is that related species often differ in pollination and mating systems. And interspecific variation in sexual traits is not unusual. When we talk about intraspecific, we mean within a species. We find that we have a variety of the variation in the mating systems. And this is very true among the herbaceous plants. And of course, then we have also a variety of mating strategies and sexual systems. Now, hermaphrodites are those which, in a single individual, they have both the male and the female structure. And this hermaphroditic condition is characteristic of angiosperm species that facilitate opportunities for both cross and self-fertilization. Now, patterns of mating in plants can therefore be both very complex and very highly promiscuous in comparison with many animal groups. Promiscuous, very high in plant reproduction systems. Now let's look at how some of them look like. The first two plants show the dioecious condition in which male and female flowers are born on separate plants, while the fourth plant exhibits the monoecy, monoecy or monacy, where male and female flowers are born on the same plant. And that is an example that we find in maize. Maize is monoecious. The third plant, as you may be able to see here, the third plant here is bisexual hermaphrodites. Notice that the monoecious one has two reproductive substructures on the same plant, but housed in separate locations along the plant. The bisexual is where you have both the male and the female being housed in the same inflorescence. So we can describe the monoecious as bipositional. The stamens and pistils are found as separate flowers on the same plant, different houses. Now we get even into more complications with regard to the morphologies of the positioning of the reproductive structures. Here we have the subdioecy, where you can see that the pink flower is female, blue flower is male. Bicolored flower, bisexual, hermaphrodite flower. The first plant is gynomonoecious. It bears both the hermaphrodite and the female flowers. here. The second plant is Monoecious. Gyno, female, bearing both hermaphrodite and female flowers. Andro, like Andrew, the male, Monoecious, bears both the male and the hermaphrodite flowers. You can see. Then the third plant here is Trimo, meaning three, to Monoecious. It bears all three types of flowers, male, female, and hermaphrodite. Therefore, when dioecious, male or staminate flowers are found on one plant, the male or staminate flowers are found on one plant. Female or pistilate flowers are on another. So dioecious plants are like human beings. Of course, we have others who are, defectively in between because they have both the male and the female parts. It's called the third gender. But in most cases, humans are more or less dioecious. But we are dealing with plants. So we have terms which are defined as incomplete flower or imperfect flower. So is a complete flower an imperfect flower? Is it? I leave it to you. But we know that the male or staminate flowers found on one plant, female or pistilate flowers on another plant, like in this species, are incomplete. Are they imperfect? Probably yes, since they do not have all the four main parts. See this? Perfect, completely coupled. But imperfect, are they incomplete? Yes, because singly staminate or singly pistilate flowers, regardless as to whether they are monoecious or dioecious, provided they lack stamens or pistils, are incomplete. Now, it's interesting again. The physical separation of reproductive parts is very elaborate in plants. In particularly unisexual flowers, as we have seen, like humans, we are unisexual. Women, gender, and male gender. Staminate and capillate flowers. And this condition is monocetic or diacetic, monoism or diaism. So we recognize here that both monoecious, like in maize, and dioecious plants have unisexual flowers. Remember, monoecious and dioecious have unisexual flowers. A dioecious condition is the most effective mechanism to prevent self-fruit. Dioecious. Many dioecious species will not set fruit unless you plant both sexes. And examples are kiwi fruit, papaya, watermelon, avocado. Can we think of other monoecious and dioecious plants? Because they do exist. There are quite a few. So if we are looking at the monaceae, this is a very common condition in wind pollinated plants, like in maize. And this condition is very common in temperate regions. Sometimes you can get self-pollination being possible, but it's less likely. Then the diocese, it's estimated that it is about 4% in angiosperms and is scattered throughout the regions, but fairly common in tropical regions and oceanic islands. In diocese, flowers are generally of small size, and invariably in diocese, it's about 100% outcrossing, but it's fairly inefficient because so much pollen is wasted in between. And diocesis is often controlled by sex chromosomes. Now, we do have various versions of the physical separation of reproductive parts. The phenomenon is known as hacogamy. We have the approach hacogamy, where we have the pin, and we'll see this a little bit later to become clear. The pin is the presentation of the stigma above the level of the anthers. The stigma is here and the anthers are down. That's pin. It's considered to be common and is associated with a large diverse fauna or flora visitors or pollinators. Then we have the reverse hacogamy, thrum. It is displayed when the stigma is recessed below and the anthers are above. This arrangement causes floral visitors to first contact the anthers before the stigma. For this reason, reverse hacogamy is believed to facilitate greater pollen export than approach hacogamy. This type of sexual organ arrangement is typically associated with moth or butterfly pollinators. You compare that with the temporal phenomenon below. Temporal means in time. Decogamy, the ripening of the stamens and pistils of a flower at different times so that self-fertilization is prevented. But contrast this, compare with homogamy. The homogamy is the simultaneous ripening of stamens and pistils of flower, ensuring self-pollination. Homogamy or homogamy is the opposite of the hacogamy and dichogamy. So let's look at the temporal separation of the sexual organs. We have one which is called protandry. Just remember Andrew, like Andrew the man. Anthers release pollen before stigma receptive. It's common in insect pollinated plants. Protandry, pro coming before. And this example can be found in geranium flower. First day flower, you get anthers released, and second day flower, you get the stigma ready. Now let's get to this idea of the thrum and the pin. The control is structural, it's physiological, it is genetic. And these pictures are very clear. Let's use the verb pinning to put a pin flower in perspective. You can consider the pin flower to have anthers pinned down versus the thrumming of the anthers above the stigma. That stigma may be homogenical or dichomically recessed. So pin the anthers down and you have a pin flower. Thrum them above and you have the stigma recessed. I hope that helps to make that distinction. So you can see an example of thrum and pin. Really, they are an example of the spatial deployment of female gametes at a flower level. You can see the happy examples, and they are common in oxalis. Oxalis is a very common flower seen during the rains, uh, even at Ubongo campus. Now we have another spatial separation of anthers and stigmas, a very interesting display of how the things oppose each other. You know, you can imagine a man facing one side, they don't want to talk to each other, and the woman facing on the other side, back to back. And so they have no sexual interest in each other. That is achogamy that is known by the terminology enantiostyle, left and right-handed flowers, more or less like repulsion positioning. Then we have the movement achogamy, stigma movement. You see, it's like chasing the chicken who is running away, cockerel chasing the chicken running away. You get the stigma movement away from the anthers. And this occurs in this species here, the Pharmacia. Then we have differences even in the morphology of the styles. We have heterostyle or heterostyle. The flower in individuals, the same species, having 2 or 3 different style lengths, with stamen length varying immensely. For example, here, an example of hycogamy, again, we have different style of stigma and correlated anther heights. As you can see, long pin, long pin. Then we have short thrum, Then we have long tristele. Then we have mid. Then we have short. Various. So di means two. Here we have two. We have sunflower long filaments with short styles. Then pin, we have short filaments and long styles. Only pollinators between different floral morphs are successful, like can be found in these species. Then three, tristele, three morphs. Again, you can see this description. Then we have polygamous flowers. Getting complicated? Yes. Here is where you have both bisexual and unisexual flowers on the same plant. We saw that. For example, what we saw before, the androdaceae, bisexual and staminate individuals in our population, then andromonaceae, bisexual and staminate flowers on the same individual, and this occurs in the phobia in Solanum species. Then we have gynodiaceae, bisexual and capellate individuals in a population. And this goes on to various kinds. As you can see, they just reveal more of the complexity. Then we have the geitonogamy, self-pollination between different flowers on the same plant. This does not happen in maize. But where it happens, then we are talking about the geitonogamic plants. There are certain species of the kind. Now, these are simply the examples of dichogamy, as we mentioned before. So when we understand that, we now want to get to looking at the issue of crossing. Because you cannot do manual crossing, manipulated crossing, and forced crossing, artificial selection, unless you know the morphology of the flower parts, whether they're the anthers, the stigma, the styles. And you have seen the various morphological options. That are important for us to know. The intent in crossing is to pollinate so as to fertilize, and if the offspring you get is your cross. Sometimes it's called your hybrid. So let us review the flower reproductive biology here. And I'll introduce a very, very, very simplified version of this so that it can save us some time.
Our topic for today is sexual reproduction in flowering plants. Let us learn about the male and female reproductive parts of flowering plants. Stamen is the male reproductive part of the flower. It consists of the anther and the filament. The anther produces pollen grains. Each matured pollen grain contains a tube cell consisting of a tube nucleus. And 2 male gametes. Carpel is the female reproductive part of the flower. Top part of the carpel is called the stigma. Stigma is supported by the style. Base part of the carpel is the ovary. Ovary contains 1 or more ovules. Each ovule has a female gamete. Flowers that contain both male and female reproductive parts are called bisexual flowers. Hibiscus and rose are some examples of bisexual flowers. Flowers that contain either male or female reproductive parts are called unisexual flowers. Papaya and watermelon are some examples of unisexual flowers. For fertilization to take place, pollen grains need to be transferred from the stamen to the stigma. If the transfer of pollen grains takes place in the same flower, then it is called self-pollination. If the transfer of pollen grains takes place from one flower to another, then it is called cross-pollination. This transfer of pollen grains is done by wind, water, insects, or animals. After the pollen grain lands on the stigma, the tube cell produces a pollen tube through the style into the ovule. The male gametes travel along with the tube nucleus through the pollen tube. After they reach the ovule, one of the male gametes fuses with the female gamete, leading to fertilization. Fertilization leads to the formation of zygote. The other male gamete fuses with the polar nuclei to eventually form endosperm. The zygote divides multiple times to form an embryo within the ovule. The endosperm provides nourishment to the embryo. Eventually, the ovule develops and turns into a seed.
Yeah. Yes! Yes!
As you can see, you can review that slide on YouTube at any time, but at least you have it here. And it basically does summarize the good part of what we need to know. Just some detail to add is that while fertilization is taking place, we have some of the very important processes which again you encounter in Mendelian genetics and in any biology book, where you have the meiosis and the mitosis. And this detail, please look at it in the slide on your own, but suffice it to recognize here is that we do have what they call two alternating generations. The prophetic, which is basically 2N, where the embryo that is developed is zygotic, which results in the seed. And gametophytic generation is where you get the progression from meiosis and mitosis to meiosis, which then puts the gametophyte generation in a haploid state, the shortest generation of the alternating or the alternation of the plant from the prophetic generation to the gametophytic generation. Remember, seed development is one of the yield formation that we have an interest in because it is the longest, it is the most useful as a source of food. So this slide here shows the differences between self-fertilizers and open fertilizers, plus the third dimension, which is asexual propagated material. Again, note the differences. And we have about 5 mechanisms that promote cross-pollination, genetically speaking. We have male sterility, very important. The pollen grains are not functional. Then we have the GMS, is caused by nuclear gene, genetic male sterility. Then we have the SMS, which is caused by cytoplasmic gene, cytoplasmic male sterility. Then we have the cytoplasmic and genetic caused by both nuclear and cytoplasm. Then we have the TMS caused by a foreign gene. Then we have the CMS caused by chemicals. So these are the various ones. We do not want to belabor the issue of the asexual reproduction, but we have quite some distinctions, as you can see in the second bullet. And we have, of course, the advantages, because here we have the same materials, more or less the clones appearing every time. The examples are the rhizomes, the tillers, the bulblets, little bulbs, the bulbils, inflorescence, vegetative bulbs, like we can see in this one's on top. Then we have the cuttings. Now, apomixis is a very interesting phenomenon, but we will stop at this point because we will want to focus on apomixis to some extent because it's a very useful phenomenon that is applied in plant breeding. For the moment, I'd like to thank you for listening to me on this aspect for now. Thank you. Cheers. Until next time.